A key challenge in understanding genetic risk can be identifying which parent a pathogenic variant came from. Besides the X and Y chromosomes, every cell in the body has 22 pairs of chromosomes, one from each parent. To determine the source of gene variants, we can use a method called parent of origin-aware genomic analysis, which looks at differences between the chromosomes using only a blood sample from the person with the variant. This method uses two main techniques: long-read sequencing and single-cell strand sequencing, also called strand-seq. Long-read sequencing is used to spot differences between the sequences of the two chromosomes inherited from each parent. It can also detect methylation of DNA at selected regions, called imprinted regions, that differ between the parental chromosomes, allowing for the identification of the parent of origin. By aligning multiple long-read DNA fragments to a reference genome, we can construct larger DNA segments with imprinted regions, revealing the parent of origin for each segment. However, if the variant in question is far from these imprinted regions, long-read sequencing alone does not provide enough information. That's where strand-seq comes in. This technique involves looking at live cells to track the forward and reverse strands that compose the DNA double helix. Cells are treated with a marker that labels new DNA strands. After the cell divides, the new DNA strands are destroyed, leaving the original strands, one from each parent. By comparing sequenced forward and reverse strands to a reference genome, it is possible to figure out which genetic variants are inherited together as a haplotype on the same chromosome, allowing us to tell the parental chromosomes apart. Typically, 20 to 50 cells are needed to get a complete picture. By combining parent of origin information from methylation patterns obtained through long-read sequencing with the haplotype details obtained with strand-seq, we can accurately determine the parental origin of each DNA segment. This combined approach allows us to map out the inheritance patterns from both parents across all chromosomes, giving a clear picture of how traits are passed down through the family and which side is at risk of carrying the pathogenic variant.